All right, hello everyone, and welcome to the first lesson in Cells and Diffusion Part 2. So we're in a new booklet now. You may have noticed that there's no video for Lesson 6, and that's just because Lesson 6 is a practical lesson, and since we don't really have a classroom, you don't have to worry about Lesson 6. The principles that are covered in that lesson are generally going to be covered in other lessons as well, so don't worry too much about that. So that means today we're going to be on lesson 7, which is cells in the microscope. So now that we've learned a lot about these organ systems and different organs and their functions, we're going to go more in depth on what makes them or what they're made up of. So as always, I'm going to ask you to pause the video now and get started on the knowledge check. Okay, so going through these knowledge check, let's go through these, and again, they're always going to be on previous lessons. Sometimes they might have a question or two on the lesson that you're going to do that day, but if you don't get those, that's totally fine. So number 1, state the organ that produces hydrochloric acid. As I mentioned before, if you ever see the word hydrochloric acid, make sure that you associate it with the stomach, okay, because that's where all the acid is produced, right? Stomach acid. Number 2, state the organ which recycles red blood cells. So again, this is a function question. This one is going to be your spleen, okay? And that's going to be right next to your stomach. Number 3, state the organ which absorbs water into the blood. This is the one where you have to be careful. It is part of your intestines, but it's figuring out which one it is, right? So the one that absorbs water is actually your large intestine, okay? Which means that the other one, which absorbs food, will be your small intestine, okay? Perfect. Number 5, suggest which organ removes urea from the blood. Now, urea kind of sounds like urine, right? And the organ that's mostly responsible for producing urine is your kidneys, right? They're the ones that filters your blood and removes the urea, which produces urine, okay? Number 6, suggest a scientific name for the gullet. So there's going to be a lot of terms that you need to know, all right, that have multiple names. So for instance, the windpipe is also named the trachea, and it also goes for some of the bones as well, such as your skull can also be referred to as your cranium. Okay, so it does help to know some of the different terms for different body parts. Number 7, state the function of the gullet. All right, so the gullet or your esophagus is that muscular tube, okay, that moves food from your mouth to your stomach. And I think I did mention this in one of the previous lessons where you don't consciously move food from your stomach, or sorry, from your mouth to your stomach. Your muscles kind of do it out of habit. And this is a process called peristalsis, if you want to look it up. And peristalsis is just that movement of muscles that pushes the food through your throat down into your stomach. Okay, which is another reason why you can actually eat food upside down and that food will still reach your stomach. Okay. Number 8, suggest which organ acts as a double pump. So that should be acts as a double pump. And this one, as you know, is your heart. Okay, so there's your do now answers. And I think I always tell you guys to make sure you have the correct answer so that when you're revising, you're not revising incorrect answers, okay? Okay, so like I said before, the majority of this lesson is on kind of the building blocks of life, which are the cells, okay? And we are going to look way more in depth on these in the following lessons. So this kind of gives you a basic overview and a little bit about how we got to this point, okay? So obviously we're going to talk about microscopes a bit today. So let's start off by reading off this section right here. It says the body is constructed of cells, tissues, organs, and systems, and these structures are placed into a hierarchical system as shown below. So let's first highlight this word right here, hierarchical, okay? And it comes from the term hierarchy, which you might have heard in another lesson. I know some of my students have heard this term before in a different subject. And a hierarchy is essentially, it's a system of order, okay? And in this hierarchy that we're going to talk about today, generally goes from smallest to largest, but a hierarchy can be made up of different things, right? In a business, okay, or a company, the hierarchy goes, you know, you have the CEO, and then you might have regional managers, and then you have other managers, and managers look over employees. And so that's kind of like a hierarchy of who's most important, right? Down to the least important, or maybe who makes the most money. And in other lessons, you might have heard it in a medieval sense, right? In history, where a hierarchy, at the top of the hierarchy, you would have the king. And under the king, you might have lords or, you know, something of that sort. And at the very bottom of the hierarchy, you have the peasants, right? So that's a hierarchy, all right? There's an order of things, a system of order. So your teacher will give you an example using heart muscles leading to the circulatory system. Okay, write this on this page right here as you are shown. So what I'm going to do is, again, I'm going to make these annotations in red so that you know exactly what you have to put in your booklets. All right. So we're going to start with the heart muscle cells. Okay. So that's the first part here, is you have the cells. Now, if you have a bunch of cells together, they're going to create tissues, all right? Not the tissues that you blow your nose on, but these are tissues that are made out of cells, okay? And when you have a bunch of heart muscle cells together, I'm going to use kind of the medical term, and that's going to call that cardiac tissue, okay? Cardiac, it refers to your heart, okay? That's why we call it like cardiac arrest, right? If you have a heart attack, that sort of stuff. And if you have a bunch of tissues together, okay, you're going to create an organ. And when you think about heart muscle cells and cardiac tissue, the organ that we're talking about is your heart, okay? And when the heart is part of a system, okay? So notice how we're going up in size here, right? Cells are the smallest, and we get a little bit bigger into cardiac tissue, get a little bit bigger into the heart, okay? And then the system that the heart is in is going to be your circulatory system, okay? Your circulatory system. Now, the organism that has this, now this, you can go into different, I guess, organisms, because lots of living organisms have hearts, but we're going to talk about humans, right? Humans are part of your, well, sorry, humans have circulatory systems, right? But you can do the same thing with, you know, like a cat or a dog or any sort of animal, right? If that animal has a heart, because all animals have a circulatory system. Okay, so this organism part is just basically what living thing are we talking about right now. All right, so you can do this with, you know, almost any living organism, and they can be broken down into cells. Okay, so if you scroll down here, you'll notice that there's a big chart with a bunch of definition. And what I want you to do now is, again, if you're working on a digital copy, you can just highlight this part right here, because I want you to recognize that this is actually quite an important section here. Okay, if you're using a paperback copy, you can highlight all this, or you can just put a star to the side here just to signify that this is an important section. Okay. So just to kind of recap this section right here, so as I mentioned before, an organism, right, is that living thing that you're looking at. It can be a sort of dog, a sort of cat, whatever. Sorry, I keep using the same examples over and over again, but you kind of get what I'm saying. So if you look at the very, very basic parts of a living organism, all right, they're made up of cells, which are the basic building blocks which make up all living organisms, right? All living organisms are made up of cells. There are different types of cells, but, you know, at the base of it, they're all cells, okay? When you have a bunch of cells together, so look at the definition of tissues, a group of similar cells that work together to carry out a particular function, okay? So when you have a bunch of these cells together, and the important part is that they have to be similar. You can't have heart tissue and muscle tissue and stomach tissue in one place and expect them to create an organ, right? Because organs have a very specific job, okay? Your heart cannot breathe for you, right? Your heart can't do the same things that your lungs can do, which means that your lungs and your heart are made up of different kinds of cells and tissues, okay? So remember that they have to be similar cells, and then you get organs, okay? Now, here's where it gets a little bit more complicated because an organ system has a bunch of different organs working together. Okay, so in your systems, let's take your digestive system for example. Okay, your digestive system is made up of a lot of different organs, all right, such as your stomach, your kidney, or sorry, your stomach, your liver, your pancreas, your large and small intestines, and all of them are made up of different types of tissues, right? Because your stomach tissue is going to be different from your small and large intestine tissue. Okay, but they're different organs, right? But they both, all the organs in your digestive system all work towards a particular function, and that's to process and digest your food. Okay, and the organism that we're looking at is all these different organ systems working together. So now you have, your circulatory system, your digestive system, your respiratory system, all these different systems working together to keep this organism alive. Because if you didn't have any one of your organ systems, you wouldn't be able to survive, because each one of your organ systems is responsible for doing a particular sort of life process. Okay, so think about the 7 life processes, and you can fit all 7 of them, okay, into different organ systems in your body. So for example, your digestive system is responsible for nutrition. Okay, your respiratory system is responsible for respiration. All right, and your muscular system, okay, and your skeletal system actually are both responsible for movement. So all of these things are very, very important to maintaining and making sure that you do all 7 of those life processes. Okay. So now we're going to look at this little big paragraph right here. And I'm going to try and make, as always, I'm going to make annotations, and if you need to pause the video just to write them down, sometimes they might be a little bit longer than what I expect it to be. So pause the video whenever you need to in order to make sure that you have the proper annotations on the side here. Okay. So I'm going to start off in line 4. So far we've looked at structures visible to us with the naked eye. Okay. We've looked at organs and organ systems and organisms, which we can all see if I was to cut myself open. The systems and organs of the body. We are now going to look at the microscopic world of the cell. And I want you to highlight this next sentence here. Cells are tiny building blocks of life, and they are made up of even smaller structures, such as the membrane, the cytoplasm, and nucleus. All right. Now the word that I'm going to introduce here is organelles. Okay, and organelles are literally called, well, they translate into tiny organs, which means that just like how we're made up of organs and cells and tissues, okay, cells also have their own quote-unquote organs, all right? But they're not the same like a heart or a brain or lungs, but each organelle has a very important job to do. So a membrane has a job to do, a cytoplasm has a job, the nucleus has a job, and we're going to look into those organelles next lesson. So in order to study these cells, we need to see them, and to do this, we need to use a microscope. And a microscope is a piece of equipment that allows us to magnify an object so that it can be seen. Okay, so again, I want you to highlight this sentence right here. And without the microscope, we would never learn about, the extremely small life in the world that's invisible to the naked eye. Right? Because if we didn't have it, we wouldn't know about cells. We wouldn't know about tissues. Okay? And so it's actually really important that we have these microscopes so that we can actually see why our body behaves the way that it behaves. Okay? And so you can check out this link here on BBC Bite Size that tells you a little bit more about microscopes and how they work and about the different ones. So if we were in a classroom, I would definitely show you these light microscopes. All right, and they use, like the name implies, they use light and lenses to form a magnified image of a specimen. It has some basic parts, including an eyepiece lens, objective lens, stage, coarse and fine focuses, clips, and handles. So look at this diagram right here. Okay. And what I'm going to do is I'm going to go through each one and define what they are, or kind of what they do. And that way, you know exactly, if I ask you, what is the purpose of an eyepiece? What is the purpose of an objective lens? You can tell me exactly what that purpose is. Why do we need it? Okay. So what I want you to do is, as I go through these annotations, all right, I want you to put them on the side here and I'm not going to keep them up. So again, if you need to pause the video, just feel free to do that. All right, that way you can get all the annotations onto the side here. All right, so the first part that we're going to talk about is the eyepiece, and the eyepiece is where you actually look through. Okay, the interesting part about the eyepiece is that it has another piece of magnifying lens inside that most students forget about. They only think about the objective lens, and that's kind of, it's dangerous to think that, because sometimes you might look at a picture and you don't take into account that the eyepiece actually has a piece of magnifying lens in there. And sorry, I should explain that magnifying lens are kind of like glasses or a magnifying glass. They're a piece of glass that will enlarge an image so that you can actually see smaller parts of it. Okay, so that's your eyepiece. Your objective lens is going to be the most important part on the microscope, because this is where you can change the amount that you're going to focus in to your whatever you're looking at. So the objective lens generally has 3 different lens. Okay, some of the fancy ones will have 4, but at our school we have the ones that only have 3. And generally, the objective lens will have numbers on them. One will say times 4, one will say times 10, and one will say times 40. And what these numbers represent is, the numbers represent how many times it's going to enlarge the image that you're looking at. So if you have 4 times, it's going to enlarge the image 4 times bigger than what you would actually see. Same thing for 10 and 40. It will enlarge it 10 times bigger than what you will actually see. Okay, and normally you will be able to, with your hands, you can switch around the objective lens by just turning it around. Okay. All right, on the stage, so the next part is the stage, and just like, you know, in theater and whatnot, the stage is where you prepare and you see everything. So the stage is going to be where you put your specimen. Okay, and the specimen is what you're looking at. That's generally what we call, you know, what your object is. Okay, so the stage is where the specimen is placed, and it also holds onto the slide. So if you see these 2 little yellow rods here, these are the clips. Okay, it's not labeled, but these are the clips. And the clips will hold on to the slide. All right, and that kind of brings me to the next one, the slide, which is a very, very thin piece of glass where the specimen is prepared. So for instance, if you wanted to look at an onion cell, right? If you wanted to see what the cells of an onion look like, you would cut a piece of onion, okay, very, very thinly, and you would put it onto the slide. Okay, and the slide is made out of glass so that it doesn't obstruct the light coming in. All right, so on the stage there will be a hole where the light can actually shine through. Okay, and so this next part here, the light, is actually the key point of the light microscope. All right, you have to have light in order to use the light microscope, or else, you know, you can't see anything. So that's where the light's going to come out from. Okay, really quick fun bit here. The very, very early light microscopes, actually, this part didn't have a light bulb. You actually used a mirror. Okay, and so you needed to be in a very, very brightly lit room in order to use a light microscope, because the microscope would use a mirror to shine light through the stage. And nowadays you can just plug in a light microscope into an outlet, and now we have light bulbs. That just raises the cost of the light microscope a bit more, but just so you know. Okay, and then the last 2 bits here that are labeled is the coarse and the fine focus. Okay, and these 2 buttons will help you focus on the image that you're looking at. So kind of like how your phone, or if you have a camera, right? Most of the times the camera will autofocus it for you. You don't have to move. The camera will actually move itself, move the lens in order to focus on the image that you're trying to take a picture of. On a microscope, you have to do that yourself. So these are the manual knobs that will help you focus. Fine focus is very similar to fine tuning. So if you want to focus on an image, this will actually move the stage in very, very small increments. Okay, so it actually moves the stage up and down, very, very small. Whereas the coarse focus, this will move the stage a little bit farther up and down, because if you're looking at something that's quite large in terms of microscopic behaviors, you'll notice that you need to move the stage quite a bit in order to focus it. So you would start with the coarse focus first until you get a nice enough image, and then you can use the fine focus to really fine tune what you're trying to see here. Okay, so it's your coarse and your fine focus. Word of caution here is that some microscopes will not have both of these. Okay, some microscopes only have one focus, and that's just because that's the way that they were manufactured. But that doesn't mean that the microscope is any less just because it only has one. Right? That just means you have to be a little bit more careful. And the last two things that I want to mention, because normally in a practical where I have you handle these microscopes, I would explain two more things. Okay, and one of that is the arm. Okay, the arm and the base. So the arm is this section right here. Okay, and when you are carrying a microscope, you would always have one hand holding the arm and one hand on the base. And this is just to ensure that you're not swinging around the microscope and you're not holding it by one hand, right? Because these microscopes are not cheap. And you need to handle these microscopes with a lot of care. So you would have one hand on the arm, one hand on the base, and that's how you would carry them from where they're stored to the table that you're working at. Okay, so just make sure that you label these two parts. Okay, so that's your arm and that's your base. Okay, and those are the parts of the microscope. And if you scroll down here, you'll notice that it's the same picture with the same arrows, but it's all blanked out. So what you want to do now is pause the video, but do not look back at the previous page. Okay, don't look at the back of the previous page, and try and fill out as much as you can from memory, because as always, when you're working with the microscope, if you're in a practical and the teacher says change to a ×4 objective lens, okay, you need to know what an objective lens. You don't. It just makes it a lot faster if you don't need to grab your booklet and try and find what an objective lens is, or what the stage is, or what a slide is, right? So pause the video now and make sure that you can fill these out without looking back, okay? And once you're finished there, you can scroll back up to the previous page and use this right here to make sure that you have the correct answers for this image right here. And as always, if you had the arm and the base on there as well, that's perfect. But as long as you have it on the notes page, that's fine by me. Okay. Before I move on to the next type of microscope, I just wanted to show you really quick some images of what a light microscope, what you might see through a light microscope. So if you go into Google and just type in light microscope images, this is what you're going to come up with. And you'll notice that a lot of these pictures are really colorful. Okay, now I don't want to kind of burst your bubble, but generally when you actually look through a microscope, it's not going to be like this. Okay, and that's just because a lot of these are what we call stained. When the slides are prepared with your specimen, whatever you're looking at, we will generally add a certain chemical to stain the cells. Okay, and we do that because we want to actually see different parts of the cell. If we didn't stain it, you wouldn't actually be able to see a lot of the details, because many of the cells are very clear, okay, which means that they're see-through. So it's kind of like if you shine your light through a mirror, sorry, not a mirror, a piece of glass, right? And you're trying to look for an image on the other side, but you won't see anything because it's just made out of glass. So, but if you color that glass, then the light that shines through will turn into that color. And so just by looking at some of these, so this is an onion cell right here. Just by looking at this, the stain that you use has a purple color, okay? And you can see each individual cell, okay? So where the lines are, that's each cell, and the little dots inside, those are actually the nucleus, okay? So there's a reason why we stain certain things. And if you go through, this whole bunch of photos here, all right, this here, that's a mitochondria right there. And so different stains will attach itself to different organelles, depending on what stain you're using. And that way you can actually identify, well, this cell has a mitochondria. Okay, so these are some images that you can see with a light microscope. Okay, this is actually an electron microscope, but you can actually see mitochondria with a light microscope, just not this much detail. And I'll explain that in a little bit here why. All right. So here we go. The electron microscope. All right. So sometimes the light microscope does not allow us to look at all objects in enough detail. All right. An electron microscope uses electrons instead of light to form an image. Okay. So here's something I want you to highlight. Okay. So an electron microscope is actually a lot more powerful than a light microscope. Okay, and the way that electron microscope, obviously you don't need to know the details of it, but what you should know is that if you think back to your atomic structure, okay, atoms are made out of protons, neutrons, and electrons. Okay, and what we can do, we actually have the technology for this, is you can shoot a bunch of electrons through a cell, okay, and what they'll do is they'll bounce off of certain materials, and electron microscopes are really cool for that because sometimes you can create a 3D image of a cell, and I'll show you a picture of what I mean. Okay, and depending on the type of electron microscope, you either get a 3D image or a 2D image in a lot more detail. All right, so going along, they have a much higher magnification and resolution. So I want you to highlight this section right here because this is a definition that you need to know. Okay, this is very important. Resolution is the ability to distinguish between 2 points. So a higher resolution gives a much sharper image. All right. The best way that I explain this to my students is with TVs. All right. Resolution is basically how well you can see a clear image. So if you compare TVs from 20 years ago to the TVs that we have today, the images and the videos from 20 years ago are very, very grainy. They don't have very good sharp images. Whereas if you look at the videos now and the images now, you look at them and sometimes you think, well, I can't tell if this is, computer generated or if it's real life. Okay, so when you watch videos these days, right, even from your phone, it's almost like you're capturing real life. Whereas video cameras, phones from 15, 20 years ago, even if you recorded them, they're very grainy and it's very hard to tell sometimes unless you're looking right or unless you put your phone right in front of what you're trying to look at. So that's what I mean by high resolution. Okay, you can see if you have high resolution, you can see a lot of detail. Okay, and these types of microscopes allow us to see much smaller things in more detail. So there are some organelles in the cell that are way too small for light microscopes to see. Okay, so with the electron microscopes, I'm just going to go off in a little bit of a tangent here, and I'll show you a picture. With electron microscopes, there are some things that you cannot see. For instance, a mitochondria, okay, or mitochondria, you can see the basic shape of a mitochondria from a light microscope, but you would not be able to see these little lines here, okay? And that's because a light microscope doesn't have enough power behind it in order to see all these individual lines, okay? Same thing for like this chloroplast. You can see chloroplast with a light microscope, all right? But again, you wouldn't actually be able to see the chlorophyll inside the chloroplast, which are these little dark bands here, all right? And you don't need to know these two organelles here, the Golgi apparatus and the endoplasmic reticulum. These two are organelles inside the cell, but that would be going into a much higher level of biology. So how do we actually see these two images, right? And when you're looking at microscopes, okay, there's two major types of microscopes. There's the SEM and the TEM, which stand for Scanning Electron Microscope and the Transmission Electron Microscope. And right off the bat, you can see one of the major differences is the type of image that you get. So a scanning electron microscope will give you a 3D type of image. Okay, so you can see that these pollen grains, we're looking at pollen grains here, you can actually see what the outside of it looks like. Okay, and you know that the shape of these looks like a ball, right? Whereas if you're looking at this, you wouldn't be able to know if this pollen grain was either flat, like a disc, right? You know it's round in some shape, but you don't actually know if it's like a disc or a ball shape. So that's where scanning electron microscopes come in to be really handy, because you can actually see what sort of cells look like from the outside. Okay. The transmission electron microscope is generally almost like a light microscope in that you see a 2D image, but you'll be able to see a lot more detail. Okay, so this little black dot right here would be your nucleus of the pollen grain, but from a light microscope, all you would be able to see is this round circle here and this dot. Okay, you wouldn't be able to see all these individual circles here, which I'm assuming is what the permanent vacuoles are. Okay, and if some of these terms seems, I've never heard that before, don't worry, I'm going to go over it in another lesson here. Okay, so that's just to give you a basic idea of electron microscopes. The bottom line is that if you know that an electron microscope is used to see much greater detail of certain cells, and light microscopes are just a very basic microscope, right? If you know that much, then that's kind of where you need to be at, all right? And that's it for lesson 7. Very short, very simple, okay? And what I want you to do is pause the video again to get started on the knowledge and application questions at the end of the lesson here. All right, so 14 questions in total, and some of these questions you might have had a bit of trouble with because we didn't actually do a practical with the microscopes, but that's completely fine. That gives us this time to figure out, where I can explain what would have happened in an actual practical, okay? So let's start off with number 1. List these structures in order. So I'm just going to go smallest to biggest because that's how we learned it. And the very, very smallest out of these 5 things is a cell, okay? And when you have a bunch of cells together, all right, you're going to create tissue. And when you have a bunch of tissues together, you're going to create an organ. And when you have a bunch of different organs working together for a particular function, you're going to have an organ system. And last thing is when you have a bunch of organ systems working together, then you're going to have an organism. Okay, so there you go. All right, state the definition of an organ. All right, so this organ right here, right in the middle, what is the definition? What's the function of an organ? And so what that is, it's a group of tissues working together for a particular function. Okay. Number 3, state the definition of a cell. All right, so this one, the cell is the basic building blocks of all life. Right? Every living thing is made up of cells. State the definition of a tissue. So a lot of these definitions in the middle here, tissue, organ, organ system, are going to be very similar because they kind of work off of each other. So a tissue is going to be a group of cells then working together for a particular function, right? And then the last one, what is an organism basically? An organism is, at the very basic point of view, is a group of different organ systems working together for a particular function. And that function is literally to keep you alive. Okay? All right, so what are the 2 types of microscopes? Remember that these numbers on the side here represent how many points it'd be worth on an assessment. So this question right here, obviously you'd get 1 point for each type of microscope. And you have the light microscope and electron microscope. And I just realized you can put light and electron microscope. You don't have to put microscope twice. Okay, so this one here, state the major parts of a light microscope. And there's going to be 6 parts of it because it's worth 6 points. Okay, so you have your eyepiece, your objective lens, your stage, the light, fine focus, and coarse focus. Okay, so that's 1, 2, 3, 4, 5, 6. Now you might have noticed that I didn't put slide on there because a slide is not part of a microscope. Okay, a slide is a different part where you put your specimen on it, and it's not actually attached to the the microscope itself, right? One thing that you can use here is clips, okay? And the reason why I didn't put it in the 6 main parts is because some microscopes actually don't have clips. Okay, some microscopes actually just have an inverted stage, so it's a stage that's not completely flat. It's kind of on an incline, and what you can do is you can actually just place your slide on there, okay, and it won't move around. The clips just help it stay in place. Okay, so that's why I put it in brackets here. You can use it. If I was a teacher, I would accept that as an answer, but just so you know, it's not one of the 6 major parts of a microscope. Number 8, state which objective lens you should use first when looking at a specimen. So this question you might have had trouble with because, in a practical, I would have explained this. And the answer for this is actually ×4 or lowest objective lens. And I'm going to explain this really quick because with the objective lens, the higher the magnification, okay, the longer the objective lens is. And when you are moving the stage with the coarse and the fine focus, one of the biggest reasons why microscopes break is because lots of people will move the stage into the microscope, okay, or into the objective lens, and the lens on the objective lens, okay, are very, very small, and they are very difficult to replace, and since they're made out of glass, they break very easily. So one of the major things that I would have you do as a teacher in a practical is when you grab your microscope, before you even look at anything, before you plug it in, make sure that your microscope is at 4 times, okay, which is the shortest objective lens. And that kind of makes sure that if you're moving around your stage with your fine and coarse focus, you're not going to move the stage into your objective lens, okay? All right, number 9, state where in the microscope the slide should be placed. So when you have your slide prepared and you're ready to put it onto your microscope, where are you going to put it? And that place is the stage, right? Okay. Number 10, suggest the name of the lens you look into with your eye. So when you're done putting the slide onto your microscope and you're ready to look at it, where do you actually look at? And that piece is called the eyepiece. Oops, eyepiece. Okay, number 11. State the type of microscope used to look at smaller specimens in more detail. All right, so really simple. Basically, it's asking which microscope is more powerful. Okay, and as I mentioned before, it is the electron microscope. Define the term resolution. Okay, resolution in science, not as a philosophical point of view. And the definition of resolution is the ability to distinguish between 2 different points, okay? And I know that kind of seems different from what I explained to you before, but it's in the text. And when you have 2 points on an image, all right, the difference in resolution is the ability to actually see where those 2 different points are, okay? Because if you have a very, very low resolution, those 2 different points might turn into a blur, and you wouldn't actually be able to determine that that's 2 separate points. Okay, so the higher the resolution, the better you can actually tell that you're looking at 2 different dots. Okay, but again, if you put for number 12, resolution is how well or how bad you can see a crisp or sharp image, I would probably accept that as well because you have the right idea. Okay, all right, last 2. Number 13, a bacterial cell is much smaller than a human cell. Suggest a type of microscope that would be suitable to use to study a bacterial cell and give a reason for your choice. So this one is worth 2 points. You get 1 point for saying what type of microscope, and then the other point comes from explaining your reasoning. And since it's already told you that a bacterial cell is much smaller than a human cell, I think the most obvious choice would be to go with an electron microscope. And here you would be able to explain why you would use an electron microscope, and the reason is because an electron microscope is much more powerful, right? This type of microscope is much more powerful and is able to look at very small details in high resolution. So using those terms together, one, makes you sound smarter, and two, helps you explain things in much smaller details, okay? All right, and then the last one, suggest the name of system which is made up of heart and blood vessels, okay? So key words here is the name of system, right? So whenever you're looking at a question, make sure that you always think about what the question is asking you, because too many times I've had students come and write answers, right? They look at heart and blood vessels and they think, oh, heart, okay? But they're not actually answering the question, okay? So the name of the system is your circulatory system, okay? So just please be careful. I always tell students to on an assessment, read the questions carefully, okay? Too many times the students lost points because they didn't read carefully enough. And those are your do not questions, or sorry, not your do not, your knowledge check questions, okay? So wherever you need to look at the answers, just go back on the video and pause it if you need to copy this down, okay? The last part that I'm going to show you is, this cognitive activity, because I think this is brilliant. So this image below is from an electron microscope. So from this sentence alone, you should be able to tell whatever I'm looking at is actually a very, very small photo. Okay, very, very small. Suggest what you think the image is of. So, I mean, you can think about, pause the video and think about what you wanted to guess, all right? But I'll give you the answer right now. And the answer is you're looking at an eye of a fly, okay? And I'll show you the images, what it would look like, okay? So this is where they pulled the image from, okay? And you'll notice that, yeah, if you ever look at a fly and you look at the eyes, that's kind of what it looks like, okay? Now what's cool about this is the type of image that you would get from a scanning electron microscope or an SEM, because you have that kind of 3D feeling. You know that each one of these looks very round, like a ball, okay? Whereas a TEM, or transmission electron microscope, you would only be able to see very flat 2D images, okay? So this is the eye of a fly. All right. And that's it for lesson 7. Thank you for watching.